Okay. Okay, yes, now you present me. Perfect. Yeah, I'm going to start the presentation in 2 minutes. Okay. And I think that this talk is going to be recorded. So if you're okay with that?
Yep.
Wonderful. Before working with Alice in Stanford, I was also at MIT, and I love it there in Boston.
Oh, yeah.
Yeah, it was, yeah. It was fantastic for me.
I've been here in Boston. I've been living now in Boston for just over 20 years.
20 years.
Yeah, yeah.
I only spent 2, but I fall in love with the city.
Yeah, yeah.
Este es Mateo. Este chico me hice muy amiga de él en Londres.
Ángeles, I think that we are listening to you.
Tengo el altavoz. Hola, perdón. Yeah. Sorry.
No problem.
Okay.
Yeah. Oh, go ahead, Mateo. Yep.
No, but please, now go ahead with your question, Bill.
No, I was just wondering if when you were at MIT, was that when Alice was at MIT?
Yes. Yeah. Yes. Okay. I think we can start. Well, good afternoon, everyone. Well, good afternoon if you're in Europe. Good morning if you're in America, or night if you're in Asia. It's a big pleasure to introduce to this seminar Professor Ahmad Khalil. And now I would like to talk briefly about his career. So he started in Stanford in the other coast, studying mechanical engineering, and then moved to study again mechanical engineering at MIT with Professors Roger Kam, and then doing his PhD with Angela Betcher. And finally, he decided to join the James Collins Labs in BU and start in the field of the beautiful field of synthetic biology. Also, I think that his designs, his research interests are aligned to understanding and building the biological principles and the science that life uses, and also very cool methodology as the Volver, that is a platform, a robotic platform to perform directed evolution in maybe more than 96 different conditions at the same time in the same plate. The first one thing that I would like to highlight is I don't know how many hours did you sleep during your postdoc with James Collins, because you publish a lot. So that was impressive. You know, I went through the literature, your papers, and well, you have an awesome CV. And thank you very much for being so kind to give us, to talk about your research for us. Thank you so much. And well, the stage is yours. Wherever you want.
Thanks, Mateo. Here, let me just, before I do my full thanks, let me check my slides here. Does that all look okay? Yeah. Okay, I'm going to assume that's good. Great. Mateo, thank you so much for that kind introduction. And I want to thank Naomi, Tom, Mateo, Claudia, the whole group over at Imperial for the invitation to present. Is everything okay? Okay. For the opportunity to present today. Thank you for spending your late afternoon on the UK time with me. Little known fact, I actually owe a lot of gratitude to Tom Ellis, who Mateo was discussing sort of my postdoctoral career in Jim Collins' lab, and Tom was actually a really instrumental figure in the lab and actually helping me get booted up with a lot of yeast genetics, yeast biology, and yeast synthetic biology. So a lot of thanks to Tom for that. Great. So actually, the first thing I will do today is just say how privileged I am to work with just a fantastic group of scientists. Some of you will, of course, recognize Will Shaw here, and maybe some of these other folks. This is our group here on the Charles River that basically connects Boston and Cambridge. So we're fascinated with the process of gene regulation, this process that basically all living systems rely on. And since the discovery of gene regulation, over 60 years ago, we've come to appreciate that it's a process that's controlled by complex circuits of interacting molecules, such as transcription circuits made up of transcription factors and cis-regulatory elements. We know these circuits exist basically to integrate and process signals into gene expression output states that underlie many cell phenotypes and behaviors. And I'd say, like, very broadly speaking, we're interested in deciphering kind of general principles of their design. And I want to say just also, these systems that control this process are fascinating and important for many reasons. And let me just kind of give you like a little flavor of that, just in a nutshell, why we're so fascinated with them. First, of course, these molecular circuits are the source of extraordinary levels of biological innovation. So here's one example. For example, transcription circuits are known to be rapidly rewired during evolution, and that this rewiring is responsible for producing new morphologies, for example. Right, so here's a famous example from stickleback fish where a regulatory change controlling a transcription factor, the expression of a transcription factor, is thought to be responsible for the evolution of, basically skeletal changes in stickleback fish. Yet at the same time, the components of these circuits, transcription factor circuits in particular, are, highly and deeply conserved across eukaryotes. This is just basically a map of the conservation of sequence of transcription factors across eukaryotic organisms. So it leads to this really fascinating interplay between, in a way, conservation, innovation, robustness, plasticity that we've become very interested in. This kind of somewhat mysterious thing from a design perspective, but of course, this interplay that's critical to our understanding of biology, critical to our understanding of disease. And of course, if we can understand it more, then it's of course, potentially a powerful platform and force by which to engineer biological systems. And it's certainly not a perfect analogy, but if you think about this kind of interplay in terms of man-made technologies, you can think of, for example, using ancient discrete components like vacuum tubes, assembling them in interesting ways to innovate some of our most powerful and complex, technologies today. So how do we study this problem? How do we study these circuits? This is a synthetic biology talk, right? So I don't have to motivate this extensively. Our approach is to use rational design. We use synthetic biology to dissect these molecular circuits. In this approach, what we effectively do is build from the bottom up synthetic circuits that recapitulate design features or functions from nature. We install them into cells and analyze their function. When possible, we like to use yeast, in particular budding yeast, as a model. It has conserved pathways of gene regulation, so that helps us to think about generalizability. And we sort of think about this as a reconstitution effort, like biochemists out there, right, do reconstitution of mechanism or function. This is a way of doing functional reconstitution, but at the level of circuits. So over the years, we've asked a number of fundamental questions. We've used this approach to study the circuits that control transcription and even epigenetic memory that propagate these gene expression states over, long time frames. Okay, so let's see. Right, here we go. So more recently, we've become really interested in kind of taking some of these ideas and the design principles that we've learned and actually translating them into platforms for basically engineering and applying circuits to program biomedically useful cellular functions. In particular, I'm actually going to start this talk here kind of in this area, which is, as I said, sort of a newer area in the lab, thinking about the translation of synthetic circuits, in particular for therapeutic and biomedical applications. All right. So this first, story is really a highly collaborative project that we started many years ago. It was fundamentally led by two fantastic researchers, Divya Israni, who's a former graduate student, and Huishan Li, a joint postdoc between my lab and Wilson Wong's lab. And of course, we've had this amazing collaboration with a number of faculty, including Wilson Wong, Cole Roybal at UCSF, Keith Jung at Harvard. I think I don't have to sort of tell you that we've really entered an exciting era of medicine, of gene and cell-based therapies, which are very much poised to revolutionize medicine, in my opinion. The poster child, for some of these, of course, is the CAR T cell. This is the idea of taking patient-derived immune, T cells, engineering them to express chimeric antigen receptors, basically synthetic signaling receptors that can redirect them to target and then kill cancer cells. And this approach, of course, has shown tremendous promise in many blood cancers like B-cell malignancies, but of course have faced, and this has been documented by many great review papers and other areas, face roadblocks, right, and limitations. I'm listing just a few of the kind of obvious and ones that have been sort of documented. This includes, lack of tumor-specific antigens for good specificity, lacking the controlling of T-cell activity and getting overactivity, lack of persistent activity, et cetera, right, and of course the suppressive microenvironment of tumors. These challenges and limitations have motivated this kind of bigger idea in synthetic biology that maybe we can begin to develop and apply genetic circuits to augment the behavior of these, therapeutic cells like T cells, and start to address some of these challenges. But of course, right, to do this requires, the building blocks of these circuits, right? And here in particular, for example, gene regulators, transcription regulators that can be sort of the basis of the construction of these types of circuits. And I'd like to sort of make the claim that in general, we lack gene circuit engineering platforms and parts, if you will, that are simultaneously effective from the standpoint of being a versatile platform for rapidly designing and building circuits, the things that we value in synthetic biology, but also from the standpoint of being potentially clinically suitable, right? That can be translated, into patients, for example. And my lab in just the last several years has really tried to, make contributions to the space that can kind of start to bring these to, effectively increase this Venn diagram and build platforms for gene regulation control that can be used for these applications. And I want to just share with you at least kind of one way in which we've stepped in this direction. So recently we developed a set of transcriptional regulators, which we call synzifters. Very briefly, these are compact humanized synthetic regulators based on zinc finger domains. Zinc finger domains are really privileged scaffolds. They're small. They're the most prevalent human-derived DNA binding domains found in natural transcription factors. They can be reprogrammed to target new sequences and then concatenated to recognize longer sequences. And there's been, of course, extensive work dating back many years that have tested long-term clinical viability of zinc fingers in even non-human primates and shown really good safety profiles. So working with our collaborator, Keith Jung, we set out to design a set of 6-finger proteins whose target sequences effectively minimize identity with the human genome as much as possible. So effectively trying to make them as distant as possible from human genome sequences. And then kind of use these, this collection of zinc fingers to build these synzifters, these transcriptional regulators, by fusing them to human, transcriptional effector domains. So these are, I think, an interesting set of parts for mammalian synthetic biology. They have these nice properties that I was sort of alluding to before. Maximize the use of human proteins, and this is important for reducing immunogenicity of these constructs. They're programmable and quote unquote orthogonal, and I'm going to show you right now they can be safely and tunably controlled. And they're quite small, so they can actually be encoded into standard viral and other vectors for delivery. So I don't have the time to tell you sort of how we constructed these, how we characterize these. You can check out the preprint, and we've kind of been in a year-long process of revising this paper. But what I would like to do is focus on how we're using these to enact new forms of gene regulation control in primary human cell types, the ones that we're really interested in targeting, and in particular in T cells as a proof of principle. And the basic question is, could we establish multi-input, multi-output control with these, focusing on user control? And this is for two reasons, really. I think user control has been kind of lagged behind some of the amazing technologies that have been developed, like synthetic receptors that enable cell autonomous control. And as I'm going to show you, user control has this potential for enacting really precise and dynamic control. And the concept that we're really interested in is because of, to address complex diseases like solid tumors and autoimmunity and other things, we're beginning to sort of appreciate that what we really need are therapeutic constructs that can mount multiple therapeutically relevant actions, not just one, right? So for example, for CAR T cells, promoting T cell responses by combating immune suppression, promoting persistence, preserving the stemness of T cells, etc. And by enacting user control, in principle, you have the ability to control the magnitude of these actions, as well as the timing and sequence of these interactions. A concept that I think is very interesting. Okay, so how do we realize this? Using these synzifters as a basic kind of unit or platform, we constructed a set of genetic switches that can be activated or controlled by small molecules. And the key idea here was to prioritize the use of small molecules that are either already clinically approved, right, as drugs, for example, or are known to have favorable safety profiles. The idea being that we could then control gene expression post-delivery using, for example, FDA-approved drugs. And so here's two examples of these, orthogonal switches. This is one that's controlled by Grazoprevir, an FDA-approved antiviral drug that has an exceptional safety profile. It's taken by patients at high dose for long periods of time. So the idea here is addition of this drug stabilizes a self-cleaving protease that then activates a transcription factor, right, a synzifter construct. In the absence of this, you get cleavage and then loss of activity. Another example is this tamoxifen switch. This is kind of the well-known breast cancer drug that can control the nuclear translocation of a synthetic transcription factor that's fused to this basically evolved or enhanced version of the estrogen receptor. It wasn't, entirely obvious to us that these systems would kind of enact, or provide for, activity, gene expression activity that's relevant in vivo and that could be kind of clinically relevant outputs, right? So to test this as a proof of principle, we established a gene switch that controls a chimeric antigen receptor, in this case, the well-known HER2 antigen chimeric antigen receptor. Now, a gene switch for a CAR is interesting for many reasons. Groups such as Crystal Mackles and Cole Roybals and many have shown that controlling the timing and level over CAR activity is useful for preventing things like tonic signaling and preventing exhaustion of T cells, right? Okay, so here I want to just show you some data from building these constructs and then basically transducing primary T cells with them. Here's in vitro data. So this is showing you drug-dependent, nice expression of the chimeric antigen receptor to levels that are comparable with, for example, a constitutively expressed CAR, low leakage. Here, when you co-incubate your CAR, gene switch T cells with leukemia cells that are expressing the HER2 antigen, you can see here in vitro very nice drug-dependent, Grazoprevir-dependent killing of the tumor cells. And then in a basic, in vivo study, where we challenge the animals with these leukemia cells, and then administer the engineered T cells and administer drug or no drug, you can see here really nice control of tumor growth in mice that receive the T cells and the drug. Great. All right. So immune cell function is, of course, regulated in a complex ways, right? By communication molecules like cytokines, and generally this is in a dose-dependent fashion. Cytokines are, of course, really interesting, and people have, been interested in using them as immunotherapeutics and dosing patients with them. But of course, they often lead to severe side effects because they're very dose-dependent. So tight, control and tuning is really needed for cytokines. So to establish this, what we did was developed a Synzifter switch whereby the second drug, tamoxifen, controls the expression of an engineered variant of IL-2, a proliferative cytokine as a payload, right? To explore the feasibility of whether we can make basically an on-demand proliferation switch. So let me show you that same data here in vitro. This is measuring super IL-2 secretion, where you can see here really nice secretion levels when you administer with the drug. If you just culture primary T cells in the absence of IL-2 in the lab, right, they basically sort of, peter out, right? They need this to sort of be proliferative and maintain. And so that actually is a really great way of testing our switch. So here you can see just like regular culture without IL-2, and then for different durations of dosing with tamoxifen, you can see sort of nice proliferation of the T cells in vitro. And then in a really beautiful experiment that Huishan did, where she infused these engineered T cells into mice, and treated them with tamoxifen, and then measured T cell numbers basically in different compartments, such as the peripheral blood and the spleen. You can see here really nice in vivo evidence of expansion or proliferation of cells in the animal. All right, let's put this all together. This is brand new data. This is a genetic program in which 2 therapeutically relevant genes are under orthogonal FDA-approved drug control. And the idea is sort of, can we encode a temporal sequence, right? A priming signal leads to sort of low-level proliferation of the T cells, effectively priming them for challenges. The activation signal would then express the chimeric antigen receptor and activate T cells. And the idea is, could this sort of concept of temporal sequence activation lead to sort of synergistic in vivo functional activity? Huishan designed an ingenious animal study to sort of test this idea. Okay, so here, let me just kind of explain this briefly to you. What she did was inject animals with these engineered T cells, but with no leukemia, so no tumors, right? And then basically wait for a week, either providing tamoxifen or not. Then a week later, challenge the animals with the tumor cells. And again, then activate or don't activate the T cells. You can see here, right, in the case with no T cells, there's rapid growth of these tumors and the animals succumb. And what you can see, I think, quite clearly is that in mice receiving the T cells and this sort of sequence combination of tamoxifen and grazoprevir, they are able to, to some degree control, the tumor growth and show clearance in a kind of a synergistic way, right? Whereas cells that are not primed are really incapable of responding to the challenge. You can see here, quite clearly in the survival curves as well. This provides evidence that we can really engineer cells in which at least 2 therapeutically relevant and synergistic payloads are being controlled by 2 FDA-approved drugs. I think a really nice and first-in-class demonstration. So let me sum up this part of the talk. We developed a collection of programmable humanized synthetic regulators we call synzifters. We use these as the basis of building genetic switches that can be controlled post-delivery by clinically relevant small molecules for this concept that I'm calling kind of multidimensional user control of cell behavior. I focused on user control, but I will just like to advertise some work that we have done separately with Cole, led by Cole Roybal's lab at UCSF, using these synzifters as the basis of building a new version of SynNotch, synthetic notch-like receptors that can enact cell autonomous control over T cells. The paper recently came out, so I encourage you to check it out if you're interested. And maybe next time I come back to Imperial, I'll tell you about all the kind of other amazing kind of work that we're doing, translating these and other genetic circuit engineering platforms into many other human cell types beyond T cells. This work is moving quite quickly. Okay. I want to sort of transition a little bit now and go back and tell you a little bit about a story that's recently completed, led by Megan Bragdon in collaboration with Nikit Patel and my long-term friend and collaborator Caleb Basher at Rice University. And this is an example of how we're using synthetic biology to study natural design principles of these circuits. I'd like to start by just saying, right, an important aspect of synthetic biology that's become relevant very recently is to sort of examine really the interface between the synthetic and the natural or the host, right? And how circuits influence cell physiology and vice versa, right? So this has been documented by many people recently, including beautiful work from Tom's lab and many other labs. And we've come to sort of appreciate that crosstalk between, of course, these synthetic circuits and host mechanisms can disrupt circuit function. Correspondingly, synthetic circuits can impose fitness costs on host organisms. And that loss of fitness can have detrimental effects, right, for the stability and application of synthetic systems. For example, right, you get basically populations of cells, that are not functional, having selective growth advantages that would slow production of your engineered cells. So let's take a step back and think about this problem from the standpoint of transcription circuits. These circuits are basically, the connections between these circuits are mediated by, in essence, the binding of transcription factors to cis-regulatory motifs, right? And we know that proper cellular function really depends on the fidelity of these interactions, right? These transcription factors must recognize gene-associated, targets while avoiding off-target, interactions that could lead to misregulation. This is important in natural biology, of course, right? Any mutations or disruptions to that, what I'll call network fidelity through, for example, mutations to transcription factors or altered expression levels, can lead to, for example, in multicellular organisms to oncogenesis. It's also, you can imagine, of course, another potential disruption are synthetic circuits, and provided basically causing a disruption that can lead to sort of these types of misregulations as well. This concept motivates us to sort of think about and examine the fitness costs of our zinc finger-based transcription factors, and now kind of moving into yeast because it's a great organism for being able to sort of assess the fitness consequences of genes and circuits. So here what I'm showing you is a really simple circuit where we have a small molecule controlling the expression of these are yeast-based synthetic zinc finger transcription factors that we have developed many years ago, even dating back to my postdoc days and being used by many labs. Upon expression of these, it activates a reporter. And what you can see, I think, quite clearly here is that when this circuit is induced, you get activation, but then you get, of course, if you measure this over multiple days, you get a concomitant loss of cellular fitness. And this is not specific to a particular zinc finger. This is generalizable to others. So how do cells establish circuit connections to maintain network fidelity, right? If we look at natural systems, we see that the strategies for making these connections actually vary across organisms and across phylogeny quite drastically. So this picture that I'm showing you here, which we used in the first kind of chapter of this talk, is very much a prokaryotic-like mechanism where in bacteria, for example, these networks are maintained by transcription factors that basically have what I'll call high information content, okay? In other words, they bind large footprint, motifs with high affinity interactions. They're capable of addressing unique sequences in a small genome. This is the information content of a bacterial genome. In contrast, in eukaryotes, right, we see basically evidence for transcription factors that have very low information content. In other words, they bind really short and often highly degenerate motifs, right, that are incapable of specifying unique sites. So how is this possible? How are specific connections, achieved? This is, of course, an active area of research, but one dominant hypothesis is that this is established through combinatorial and cooperative regulation, right? Indeed, we know eukaryotic transcription factors, universally property of them is to display cooperative binding on DNA at clustered motifs. And we all know that, right, to a first approximation, cooperativity is this really interesting mechanism by which components can do more together than they can independently, as other parents of twins know very well. And in fact, if we look at natural, eukaryotic promoters and enhancers, we actually see even like the presence of multivalent complexes of transcription factors and cofactors. And yep, surprise, I actually have 4 kids. So it's so this really suggests, that specific connections are established and reinforced potentially through these multivalent associations between weakly interacting, low specificity, basic components. So let's study this a little more and potentially test it. You can just use, a basic mathematical model to just kind of gain some intuition from this. So these are these, thermodynamic models of gene regulation, which allow you to calculate basically, binding and transcription based on, equilibrium considerations and basically concentrations and affinities of these components. And so in this case where we have a transcription factor that can bind, let's say that can regulate a target synthetic locus and also target and regulate off-target native loci, where what I'm going to show you is how you can alter these 2 parameters in particular, the transcription factor affinity and the level of cooperativity, which you can see from this basic model very clearly is as you lower the affinity of the transcription factor and increase cooperativity with adjacent transcription factors, you can effectively increase the space of on-target versus off-target regulation. Great. So can we test this idea experimentally that cooperative assembly may be a mechanism for, in fact, improving the specificity of regulatory connections in the experimental framework that I started at the beginning with? So here's our circuit, platform that I showed you before. Here's our yeast cell. We have a small molecule that controls the expression of these zinc finger-based transcription factors. And to them, we're going to append these PDZ short peptide ligands. And these will regulate basically a reporter locus. In our framework, which we've developed over many years now, we have a strategy for effectively just systematically lowering the affinity to DNA by mutagenizing a series of arginine residues in the zinc finger backbone. So you can see here, you can kind of step down the affinity and the activity of these transcription factors to sort of very lowly or weakly activating ones, weakly binding ones. And in this framework for building cooperative transcription factor assemblies that we reported on a couple of years ago, we also express a cofactor, what we call a clamp protein. And this clamp protein mediates cooperative interactions between synthetic transcription factors. And it does this through protein-protein interactions between these PDZ domains and these C-terminal ligands. Okay? And I think the thing that you just really need to know is we have tunable control over all the affinities and the size of the complex. And here what I'm showing you is, good evidence that by using, by increasing the complex size in a permissive, biochemical regime, you can basically increase to quite a large extent the cooperativity of the system. So let's use this framework to test our hypothesis, okay? Here's that, we constructed a number of basically clamped and non-clamped circuit configurations and then measured activation of the circuit and the fitness, right? So this is the high affinity independent transcription factors that have high activity but low fitness. If you just lower affinity by using that strategy that I described before, you can rescue fitness, but of course you're going to get loss of the circuit activity. And now if you implement this cooperativity strategy and increase the complex size, you can just step right back up into this regime where you have both high circuit output and high fitness. The hypothesis is, of course, is that these circuits are imposing these fitness costs through misregulation of the host cell transcriptional network. And so to test this, we performed RNA-seq following induction of these, I'm just going to focus on these 3 circuits that I showed before. I'll call them high affinity, low affinity, and the cooperative, just for simplicity. And I think what you can see quite clearly is that following induction, you get basically widespread misregulation of the transcriptome in the high affinity independent, transcription factor scenario. Whereas the transcriptomes, the native transcriptomes of cells harboring the low affinity and the cooperative circuits are, similar to one another and basically similar to background levels, right? Indicating that they can, rescue that native misregulation. The expectation, right, is that that misregulation would be accompanied or driven by promiscuous binding events in the genome. So we wanted to sort of test this and look at this. Megan, heroically spent almost a year or so optimizing a ChIP-seq protocol to look at synthetic transcription factor occupancy genome-wide in yeast. She did just so, spiked in samples with known, genomic DNA from Pombe to really be able to normalize properly and look across strains. So here's an example of, basically pull down a ChIP-seq profile at the reporter locus where you can see really nice enrichment of the high affinity and the cooperative transcription factors. And then if you look genome-wide, and I'll just, just kind of summarize this very briefly for you, you see a consistent picture basically of high enrichment, right, of the high affinity transcription factor, and this enrichment is basically, for the most part, reduced or lost, right, in the low affinity and the cooperative situation. Cool. So this kind of paints us a picture of sort of these molecular events or pathways underlying the fitness differences, right? Promiscuous binding leading to sort of misregulation of the transcriptional network. What we next wanted to do is investigate whether this strategy of making these circuit connections based on cooperative assembly, whether that could be used to enhance what I'll call just sort of the long-term stability of these circuits in culture. So to do this, we used basically a continuous culture platform that I'll introduce to you a little later in the talk. And then we followed the population following induction of these 3 circuits. So here's a measure of the growth rate or the fitness. And what you can see really clearly, I think, right, is that following induction, you get this rapid collapse, right, in the population growth rate of the high affinity, in contrast to the low affinity and the cooperative, which are growing nicely and happily. And then you get sort of a slow recovery of that population, presumably due to the emergence of mutations, right? If you assess kind of the circuit activity, here I'm just showing you flow cytometry plots of the synthetic transcription factor concentration and the circuit output. And here you can see clearly, right, a gradual loss of SynTF expression and the emergence of a circuit off population over time. Time is going in this direction here, right? And this is suggestive of mutations that are disabling the circuit and gaining selective growth advantage. We went and kind of, did mutational analysis of these mutants that emerged in this high affinity, continuous culture experiment. By the way, there were no mutations observed in the circuit genotype from the cooperative system. I should just say that outright. We've found a number of solutions, mutational solutions, but in general, I'll just classify them as we found many that are basically targeting the induction cassette, basically the mechanism for expressing the zinc finger transcription factor, and then actually in the cassette affecting the activity. If you put these mutations in a clean background, they are driver mutations. They restore fitness and, of course, lose circuit activity. That's very nice. And they do this by restoring very nicely, right? Restoring gene regulation, gene misregulation, right? So if you just take two examples of these mutations, introduce them into a clean background and run an RNA-seq experiment, here's again the high affinity from before, which you can see is basically you get a restoration. That's not entirely surprising, but really nice to see that basically this, profile looks basically like our cooperative, system, our cooperative circuits. All right. Let me sum up this part of the talk now. What I showed you was, and this is consistent with a lot of other work, including from Tom's lab and Chris Voigt's lab and many others, that synthetic gene circuits constructed from these zinc finger-based synthetic transcription factors can result in growth defects due to misregulation of the native network, the native transcriptional network, and that these fitness costs drive like a loss of circuits from the population. I think this kind of class or this mechanism for fitness reducing interactions that we showed you here will likely be very important when we begin and continue to try to engineer circuits and systems in eukaryotic and mammalian, eukaryotic genomes in particular that are complex. We drew inspiration from nature in the idea of basically making transcription factors weak, low specificity, and cooperative to develop a simple and generalizable strategy for building specific connections between specific connections in the circuit. I say simple and generalizable because I didn't show you any sophisticated protein engineering here, right? All we did was weaken the interaction affinity and then basically make them dependent on one another by engineering cooperative interactions. This strategy, I think, shows you a way to basically insulate circuits from aberrant misregulation, make them stable against evolutionary pressures. We think this will be useful going forward, in this broader kind of effort to make to use synthetic circuits for biotechnology applications and keeping them stable, for example, in bioreactors, or stable in the case of expansion of engineered cells for cell therapy. Okay, last part of the talk. I want to shift gears now a little bit from circuits to proteins and gene level. And if I'll just kind of start this by just asking, what if instead of innovating at the circuit level, we want to innovate at the gene level? And I think the idea of rational design of proteins, though people are making tremendous progress on, is a much harder proposition than it is in the case of circuits, where we benefit from things like intrinsic modularity of networks, of biological networks. The molecule I'm showing you here is a fragment from an antibody or an antibody. And I'm showing you this for two reasons. Well, first, unfortunately, we're in this kind of interesting era where basically everyone in the world knows how important these molecules are, right? Including our former president, who received a dose of Regeneron antibodies. The second reason I'm showing you these is because they're really a powerful example of the power of evolution in generating innovation at the molecule level, right? Antibodies are indeed easy to make for mammalian immune systems, right? Because they undergo this Darwinian maturation process in germinal centers where memory B cells, right, undergo high rates of somatic hypermutation, and then there's this competition that drives a natural selection for higher affinity antibodies, right? We're, of course, very inspired by this and want to develop platforms and technologies for performing this type of evolutionary scheme in the laboratory and doing it at scale. In recent year, we've become really interested in we're in these kind of methods of laboratory evolution, which we call continuous directed evolution, which are seeking to basically take the process of directed evolution, where you make basically libraries of mutants, do selection and replication, take this iterative discrete process and basically make it a continuous one, where this whole evolutionary process is done in a closed loop fashion, or in fact entirely in vivo. And here are two kind of leading examples by my good friends and collaborators, David Liu and Chang Liu, the PACE system, which some of you guys know about, and OrthoRep from Chang's lab. And by the way, aren't these just like totally appropriate photos of them? These are potentially very transformative technologies, but do have limitations. And I think the big one that I want to just highlight is that effectively these are powerful engines of mutagenesis, right, in cells. But in order to perform evolution, you must pair these like powerful engines with, control strategies and selection, right? Otherwise, you're basically just putting like a powerful engine into a chassis or into a system that can't really steer it, right? And so we need selection methods that can match the power of these hypermutation systems. And this allows me to sort of advertise that, our lab has been developing a number of technologies, including actual hardware technologies like the Evolver platform, which Matteo, brought up at the beginning. And this is basically a flexible continuous culture device that we developed in the lab with the goal of like allowing researchers to invent, automate, and scale, culture-based selections, right, for lab evolution. And I won't dwell on this. You're certainly welcome to sort of read about it. There's a lot of resources online about Evolver. It's an open source system, so about 50 labs across the world are actually utilizing Evolver right now and adapting it for their own methods. But I'll just say what it allows is researchers to individually control the culture selection conditions of each mini bioreactor and do this individual control across scale, right? And so our lab right now looks basically like these like Evolver farms, right? Great. So this last bit, I just want to tell you the beginnings of a story on how we're kind of thinking about using directed and lab evolution for antibodies in particular. And this is our kind of evolution team. This is a long-term and big collaboration with Chang Liu's lab at UC Irvine. It's led by actually James Angstman and Brandon Wong, and Arjun and Zach are supporting this as well. And this is one of, I just want to say to the audience, one of actually a series of continuous evolution projects that we have in the lab that are kind of again rapidly growing. So this is based on OrthoRep, the system that Arjun developed in Chang's lab several years ago. And OrthoRep, just in a nutshell, is basically a in vivo hypermutation system in yeast. It works through this plasmid system where you can encode a gene of interest, and then it has its own dedicated orthogonal DNA polymerase that replicates it. So this can drive, mutants of this can drive really high error rates on this gene of interest. And so it can be rapidly and durably mutated as you just passage cells with no consequence to sort of any host genomic mechanisms. Great. Recently, Chang's group demonstrated that you can pair OrthoRep with yeast, classic yeast surface display technology to display fragments, protein fragments like nanobodies and antibodies, and then subject them to hypermutation just because of them being encoded on OrthoRep, right? So this looks a little bit like somatic hypermutation in B cells. And then he showed that if you pair this with basically doing FACS, passaging cells, and then to hypermutate, and then FACS-based sorting with purified antigens and do cycles of this, you can effectively, within the matter of weeks, mature antibodies, and they demonstrated this with antibodies against SARS-CoV-2 receptor binding, RBD domains. in a matter of a few weeks. I think this they call this, by the way, a head. I think this is really, really interesting. But I think it has limitations, right? So number one, your antigen source needs to be purified, right, and conjugated, right, so that you can do FACS sorting. So that's sort of limiting kind of your bandwidth. And then, of course, it's dependent on FACS sorting, which is expensive. It bottlenecks your population. So even if you had a population of 10 to the 8th, let's say, you could really only, in the matter of like, sample hundreds of cells in a reasonable period of time. So let me, share with you a way in which we're trying to sort of scale this entirely. And this is a new selection technique that we are developing. And the way it works is it actually uses 2 yeast cells, right? So your antibody cell, which encodes orthorep in your evolving displayed nanobody or antibody. And then we have a second yeast strain that is displaying your target molecule, your antigen of interest, for example. That antigen strain is also expressing and displaying a selective iron binding protein. This is actually beautiful work from Balaji Rao's lab from North Carolina State that we've co-opted and used for this application. And by doing this, basically you can magnetize the yeast in culture, genetically magnetize the yeast in culture, because you can incubate them with iron oxide nanoparticles, put a magnet, and basically fish them out, right? We call this, we're not very creative, we call this WiMAX, yeast-based magnetic activated cell sorting. Great. And everything is genetically encoded, right? So you can encode your antigen, you can encode this, and so that lends itself to scalability. Here's just sort of characterization data. So we took antibodies from Chang that he evolved using a head from his paper, right, with different affinities. And then we used the WiMAX system to basically pull them down, right? And what you can see here is, in general, this is old data, so it's gotten actually much better, but in general, really nice correlation between the antibody affinity and the enrichment level, the fold level of WiMAX pull down. So this is suggestive that this system can discriminate and can select based on, for example, affinity. A cool idea here is you can actually scale this further with Evolver-like and other DIY automation that you can just build entirely in the lab. So Brandon Wong has basically automated WiMAX entirely with these robots that he's developed based on co-opting 3D printers, actually. And now you can run like dozens and dozens and dozens of these selections of both positive and negative selection, all entirely in an automated fashion. And so what have we been doing? We're just to kind of characterize this evolution system, we're kind of replaying the tape that Chang played back in his AHEAD paper. So for example, he showed in his paper the emergence of these 2 mutations in a clone, in an antibody, in a nanobody clone, that are necessary for neutralization of SARS-CoV-2. So we've been running this magnetic, WiMAX-based selection and pairing that with NGS sequencing over time of these, rounds of selection, where here you can see in this nice little animation, hopefully, kind of the emergence of, both this, we're seeing the emergence of the mutations, we're recapitulating mutations that was, previously discovered in the AHEAD paper, and of course, finding other mutations potentially of interest and beginning to characterize those. We're excited about this kind of now what I'll call just fully genetically encoded, highly scalable antibody evolution platform, where you can, again, genetically encode your antigen or your protein of interest. You can do this magnetic-based selection that is not bottlenecked, that is really cheap, right? You can do this on a matter of, cents to dollars, right? And do it in really, really high throughput. And I'll just kind of end by just putting kind of this framework that my lab has been trying to develop of both kind of rational design, right, of genetic circuits, and pairing this now with, using laboratory evolution and developing technologies for doing laboratory evolution of biomolecules and other biological entities, combining these two to sort of study basic principles of circuits and cellular systems, and also generate new systems of biomedical importance. Finally, I'll just say, in a paper that's coming out soon in collaboration with David Liu, basically I've told you about OrthoRep and work with Chang. I'm happy to report that we've adapted the evolver system to run now PACE fully continuously on evolver in a scalable way. So you can basically run parallel PACE lagoons. So for the folks in the audience that know the PACE system, you'll know that it's not an easy system to boot up. We've effectively tried to systematize and automate it entirely on the evolver. And in a forthcoming paper, we demonstrate this, what we call ePACE system, and its application to evolving new Cas9 molecules for new specificities, but increasingly now with David evolving a whole suite of other new molecules. And I think with that, I'll just end, and thank you all so much for being here and listening to the talk. I have a fantastic group of collaborators, some of which are listed here. We're a highly collaborative lab, as you can see, and we really benefit from these wonderful collaborations. Here are some of my conflicts. I want to thank my hosts and thank you all for being here. Happy to take questions if there's some time.
Yeah, thank you so much for this wonderful presentation. It was fantastic. And I was wondering if any of the listeners have any question for you. If not, I have a couple. Let's see. No one has a question? Yeah.
Hello, I'm Fan Kang from Tom Ellis Lab. Really
Hi.
amazing talk. Thank you so much. I have a small question. I think it's pretty cool, to use the FDA-approved drugs to activate T cell. But a small question is that these drugs also have their original drug function, right? So in this scenario, their original drug functions may become a kind of side effects in your design. Do you think this will be a problem in clinical application?
I don't think it'll be a problem. I think what it will. Thank you for that question. It's a great question. We prioritize these drugs that have kind of orthogonal specificities and mechanisms, right? Like you could argue, for example, why not use, so for example, the Grizaprevir drug targets basically a protein from the hepatitis C virus, right? You could argue, why not use a human-derived small molecule interacting domain? Well, the problem with that, of course, is now you're using small molecules that are interfering with cell processes, right? So these are specifically designed because we know the small molecules are safe. And secondly, we know that they target largely orthogonal mechanisms, right? So for the Grizaprevir case, that's clear, right? The tamoxifen case is interesting, right? Because what it is selective for this higher, this kind of engineered variant of the estrogen receptor, but of course it can hit the natural estrogen receptor too, and that might be an issue. So you're bringing up an issue there, right? Now, it is the other kind of issue is that if it is, for example, a woman who's on tamoxifen, right, for breast cancer, that's all of a sudden, not viable for this strategy, right? And so there are going to, it's a really good question. There are going to be use cases where they're just not appropriate or able to be used, right? But I think many where they will be able to be used, right? So it's just a case-by-case basis in my view.
Thank you so much.
Oh, yeah, you're welcome.
Another question?
I have a question, if I may. Can you hear me?
Yes, I can hear you. Thank you.
All right. So yeah, thanks for the great talk. I was also in Tom's lab. I'm not at Asimov in Cambridge, Massachusetts. And I was wondering about your cooperativity experiment. You're expressing 5 genes, right? The 4 zinc fingers in one of the examples, plus the cooperativity driving protein. Was it challenging to tune the expression of all of these genes so that they would behave in the way that they eventually did?
Such a good question. Actually, it turns out it's not. So this is actually a nice kind of consequence of using cooperativity. You can just blast your transcription factors because they're weak, right? So you can just, just to be clear, the transcription factors that I showed you, these are multivalent complexes, but they're built off of a single transcription factor species. So I'm not expressing 4 different species. It's just one species, but it is assembling in a, at the 4 binding sites, for example. And it turns out what you can do is just basically express them at relatively high levels. And because they're weak and because they reinforce one another, you don't need to be very careful about expression levels of these things, right? There's no fitness consequence when you just overexpress them. And that's, I think, a really nice feature. There are, of course, if you just even model this, there are regimes where if you have lower expression of the clamp or lower, where you can kind of lose your cooperativity, right? And then your system is, no longer functional. So I recognize and admit that those are, potential things to keep in mind. Yeah. I should also, thank you for that question. I didn't get a chance to tell you in the talk, but like the fitness cost is derived from the SynTF species. We show that in very careful control experiments that I wasn't able to, where I can tell you certainly it's because of having a DNA binding domain with a transcriptional activator and not due to any of the other components that are being expressed.
Thank you.
Yeah, thank you.
I think Tom has 2 questions.
2? Did you say I had 2? I got 1 question.
Yeah.
I got many questions.
Yeah, I saw like 2 hands.
Yeah. Okay. Well, I'm going to go with one that's slightly naughty, Mo. Hopefully this won't.
Give it to me.
Yeah. So you've had the yeast orthogonal set of zinc fingers for a while, and you and Caleb have had these programmable binding domains for a while. So what took you so long, right? So why was this study out now? Why didn't we see this kind of thing years ago? Was it just the idea, or has there been some real technicalities getting this over the line?
Yeah. Thanks, Tom. That is actually a reasonable question. The truth is we were aware of you have to measure over multiple days, right? So if you just do an experiment, do an induction experiment, right, in a single day, you're not going to see major fitness, effects, right? This was an investigation to look very carefully and quantitatively, right, by doing these pairwise fitness experiments over multiple days and really tease out those results. So that's actually the first way to answer your question, is like we really wanted to start moving in this direction of doing long-term and stability experiments. The sort of second thing is we were somewhat distracted because we worked on sort of thinking about cooperativity as a mechanism for signal processing first, right? So that story, which we published a number of years ago, really took the lead. All the while, we had known in our back, that another sort of kind of axis, right, of cooperativity is to sort of enhance specificity, and that was going to become the next paper. And so, in a way, it's kind of phasing projects, Tom, but. Yeah, I hope that's satisfying.
Don't you have enough people?
Yeah, but we have too many model organisms and too many projects.
Yeah, that's true.
Yeah.
Great talk.
Now, thanks to Will, we're in plants, right? Thanks, Tom.
Yeah, I have one question for you. I'm a big fan of directed evolution, and in the last slide you showed that you have published with collaboration with David Liu and Nature Biotech, no? About the application of robotics.
Yeah.
So in 2020, well, this year, Mike Esveld published brands that is applying robotics to pace. What are the difference between these two?
Oh, yeah, no, great question. So thank you. So what Matteo is referring to is Kevin Esvelt and colleagues published a really nice paper earlier this year or last year on a platform called Prance. It's based on commercial liquid handling robots, right? So, your Tecan or your Hamilton robot, right? And the idea was, can we program it to, effectively do not continuous, but kind of continuous, right, pace, right? And I say it's not continuous because pace is a continuous culture system where you're flowing cells into a lagoon that consists of phage, right? And it's a continuous selection system. PRANCE is not a continuous selection system, right? Because you're doing these kind of automated, discrete dilutions. So one way where it's different is, of course, well, one is continuous and one is not technically continuous. I think there's advantages and disadvantages to both. Now, so the PRANCE system also depends on you basically building it off of an expensive commercially available liquid handling robot, right? The ePace system is basically doing kind of something different, which is taking this flexible, open source, cheap system evolver and showing how you can adapt it to basically, miniaturized, fully continuous. So it's kind of going in the other direction. It's saying rather than doing big infrastructurally, it can be done with a flexible, low-cost DIY system. Yeah.
And just to, because I'm very curious, imagine that I want to buy this evolver. How cheap or how expensive can be? I think that the price can be around 100 or 40,000, something like that. How expensive can be your technology?
Yeah, good question. So Matteo, shoot me a note, because we have this entity called Finch Bio that distributes, evolver hardware for people, so for researchers who are interested. But just as a rough figure, just don't quote me, but I think we sell like the 16 vial, base system for like $11,000.
Okay, that's fantastic. Okay, let me see if there are more questions. Any questions in the audience?
I have a question.
Please go ahead.
Okay. Thanks for the amazing talk. I just have one question regarding your last part of the talk, where you use a clamp which has multiple binding sites for your low affinity transcription factors. Okay. So the clamp is essentially recognizing PDZ domains, right?
The clamp is composed of PDZ domains. These are basically.
Okay.
Yep, and it's recognizing C-terminal ligands, like 9 residue C-terminal ligands that are affixed or fused to the transcription factors. Yes, just to clarify.
Okay, and does it not recruit other proteins that also interact with PDZ domains?
Yeah, great question. So PDZ domains come from signaling signaling networks and complexes, and we're basically driving these to the nucleus, right? And so there's no potential for interactions with signaling components in the nucleus. So yeah, and we've we've shown this a number of So yeah, I mean, yes, in principle it could. They're really derived from kind of mammalian signaling components, but because they're in the nucleus, they're effectively sheltered from some of that.
I see. All right. Thank you.
Yeah, sure.
Well, I think that there are no more questions. Thank you so much for your time and for this amazing talk. And well, you have a fan of Evolver here, and well, thank you so much. We appreciate your time. Thank you.
Thanks, everybody. Thank you, Matteo, for the for hosting.
Of course, my pleasure. Okay. Bye, everyone.
Question.
One extra question.
Sure.
But I'm sorry. Is it possible to do this? It's actually not a question. I'm Sun. I will be an